See, not what I wanted. Sorry. So now we have spoken about the contrast and how it works hand in hand with the resolution. If we have no contrast, we cannot use the resolution. We spoke about sampling as well, and how if we have a low sampling resolution, then we cannot use the optical resolution either, okay? And now we're going to talk about what happens when we undersample and the artifacts that we're creating in the image. Undersampling creates aliasing artifacts. So this is a well-known thing in photography and in medical imaging and in astrology. But somehow in biology, we just pretend it's not there. And that's a pity. So some artifacts are visible. For example, pixelation. So you can see in here this very nice curve. This curve that is very smooth in the cloth is actually jagged. And this is pixelation. And this is the fact that we're digitalizing something that is curved. So we start seeing the little squares on the edges. And another way we can see the artifacts is moire. So moire is, for example, you can see here, hopefully you can see on your screen, otherwise you will see it on the PDF. Here you have some purple and green, so some colors are a bit different. And here we get a rainbow. These are moire artifacts, and in this image that is well sampled, this is undersampled and this is well sampled, you do not see this. So clearly the objective has no problem imaging this type of tissue. This is silk, so it's a very fine material, very fine details. But the digitalization creates artifacts. So other artifacts are not visible, but the information in the undersampled image is incorrect. This is only the stuff that you can see. But there's a lot of more things, and you cannot see it. So moire artifacts are just a subtype of aliasing artifacts, and they are easy to see. So this is the reason why we always talk about moire all the time, just because we can see them. So I will try to show you things, what happens when we do not see moire. So if you take a picture of your computer screen with your phone, you will likely see something like this. And moire happens when we have two superimposed structures, and they are repeated structures. For example, a computer screen is a repeated structure, and then the camera pixels, the camera on our phone is also a repeated structure. It has a structure, the camera. So when we have two superimposed structures, sample structure and the camera chip, then we get moire like this. And what you can see is that moire adds to the image patterns like these big black bars, patterns that do not exist in the sample. This is, you can say this would be the sample and this would be the camera, and we have here these big bars that do not exist, but they show up in the image. On top of that, moire is orientation dependent. Oh, I forgot to play the little movie. That's it. So you can see that as we move the camera, these patterns are moving. But as we rotate the camera, the size and the shape of the thickness of these are also changing. This is why, depending on the angle as well, so these are really flush against each other. Depending on the angle, they can go curvy like this. So this is why here we have all curves. You can see this type of moire artifacts in life everywhere. If, for example, you have a curtains that is made with fine structure and the light goes through, you may see some moire artifacts on the curtain. It's very common. Once you start seeing moire artifacts, you're going to see them everywhere. So this does not happen at Nyquist sampling. Okay, moire is rare in images of biological samples because biological samples do not have regular patterns. This is something that happens very often in photography, where we may have, for example, a brick wall. When you take a picture of a brick wall, you may have moire artifacts, or when you take a picture of someone who has like a striped shirt, for example, you may get. So I took these screenshots of this very nice video. I spoke about this before, the eye biology microscopy course, and Jeff Liefman is one of the teachers. It just happened so that I took videos of his 2 lectures. So this is one of his other lecture where he talks about sampling. And in here, in all the images that you're going to see, we are using the same objective, we have the same object, and we are increasing the sampling. So now we are really heavily undersampling the image, and that's what we see. It's a little bit difficult to guess what it is, but maybe you can guess. Anyway, now you start guessing what it is. It's a zebra. And so we have the same objective, the same object, but we're increasing the sampling. And what you can see in here is that zebra has very strange patterns. So in here, you will see in the next image the type of patterns we get here. But this type of crossing between one stripe to another never happens in nature. If you do not know it never happens in nature, you would not be able to identify this as an artifact in the image. So as you can see, when we sample a little bit more and we have smaller pixels, then this disappears. However, in here we get a new one. So we get a checkered cheek on this poor zebra. And this does not happen in nature either. And all these wavy patterns like this do not happen. And then you can see in the background in here we have a fence. And now we see all these molten like little balls on the fence. This is also not true. It's also an artifact. Now we sample even more, and the poor zebra, it looks a little bit more like a zebra, and he's got this checkered thing on the cheeks, but he's got a nice frizzy haircut, which is not true either. And then we can see the fence now. We still have all these little balls everywhere. They do not exist. So now we sample even more, and we can still see some artifacts that do not exist, and the guy still has frizzy hair. And then slowly the little balls in the background are disappearing. So we sample even better, and now we can still see a tiny bit. So only the finest details now get artifacts. And if we sample even better, now even these small artifacts disappear, and now the poor zebra is not frizzy hair anymore. So if we take one of the worst ones with the real one, you can see the difference. There's lots of artifacts that are created in the image. The same thing exactly happens in our images when we undersample. The information in undersampled images is incorrect, okay? And it's not possible to detect because we do not know what the image should look like. So depending on the scientific question, undersampling can lead to dramatic interpretation errors. You can see this image, if the idea is that we count how many zebras are in this image, no problem. If the idea is that we study the shape of the pattern on their back, we're in trouble, okay? So we are, as always, the way we set the microscope and everything is fully dependent on the scientific question. So what about samples without a regular pattern? Because even the zebra has a regular pattern. So what happens with a sample without regular pattern? Well, it's even worse because you cannot possibly detect the artifact. You don't know what they are. So here I have tried to reproduce a sample that could be, for example, an actin label or a tubulin label. So we have random lines. I created an image of random lines. And then I displayed this on my screen, and I took screenshots of it. So I have the pattern of the line, and then I have the pattern of the screen that overlay each other. And so if I zoom out, if I zoom in, I zoom out of this image, so I see all the pixelation artifacts that is very clear on the lines. And you can see that they depend on the orientation. So this one has different artifacts. It has this strange thing here compared to that one. So it depends on the orientation of the lines. But we do not have any strange thing happening except for these that we do not resolve. So they all fuse together. So clearly the lines are not like this normally. But now if we zoom out, this is what we see. So it's the same image, just zoomed out, screenshot. And you can see that at different zooms, so different type of undersampling, I get different images altogether. Here I hardly have any lines, and here I have some lines and some jagged lines and everything. And the results that you're getting depend on the orientation of the object. So it's a bit random, and you get completely different images depending on how you undersample. So the problem is worse at low magnification, so that's because we have large pixels. And the lines look completely different depending on their orientation compared to the camera, which is actually a screenshot. Okay, so this is a little bit what happens in our samples. Now I got the luck to meet Rainer Heitzmann, who is an expert in all these. He has published several papers about undersampling, and he could do a model of what happens in a sample with different artificial objects. And so in here we have the object itself. So you can see we have some lines. They have different sort of spacing between them. So we have different frequencies, and also they have different orientations. Here we have round objects with a little hole in the middle, and in here we have all sorts of things. And here we have a little bit more something that would look like our samples. Now we're imaging and we're undersampling this image, and what you can see, we can see lots of moire artifacts in these regular patterns. But if you look at the patterns that are not regular, like here, for example, so we're losing part of this resolution that we have here, and we do not see the hole inside. And then we look in here at the center, we also have very strange artifacts. This guy here is also a little bit bizarre. But when we look at the image of the small dots, so here we have the artifacts like this, and then this one, I spoke about this already. And then when we look at here, what we can see in here, if you look at what corresponds in the other image, you can clearly see that there are 3 dots, but here they are sort of fused to each other. And we could look at this guy here, it's on its own in here. But anyway, it looks elongated. And then when we look here, we can see that even if this one is far away from the others, it anyway looks fused. And when we look at these 2 dots in here, what we can see in this image, they have the same intensity, but here they don't have the same intensity. One is much dimmer than the other. So all these are aliasing artifacts that are due to the undersampling. When we look at a point cloud focal, so this would be like the modeling for a camera-based system, but if you look at the point cloud focal, it's exactly the same, except that the patterns are slightly different, basically. But otherwise, all the artifacts are still there. Okay, so when we undersampling, we create aliasing artifacts of this sort, and the information is full of wrong. The information is wrong in the image. So single point focals are very slow, and inexperienced microscopists tend to use less pixels to go faster. And this is not a good strategy. On the camera-based systems with large pixels, we integrate the light. So on the camera-based, the optical image is projected onto camera. So anyway, even if we have large pixel, we integrate all the photons inside the image. So all the signal is recorded, even if we have large pixels. But on a single point confocal, this is not the way, because the way we sample on a single point confocal is that we take a spot and then we move to another spot and we take another measurement, etc. So we jump between spots on a single point confocal. And that means we do not record in between the dots. So if we use large pixels, it means a large distance between the sampling spot. Not only we create artifacts, but also some of the real signal is not recorded at all. Okay? So it's even worse on a single point confocal to undersample. So for example, in here, we sample along the blue line, so we're going to take dot, 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 right? And now we are sampling only at this distance. So we're sampling where we have the red things. And this is our sample, it's green. And it has a certain orientation compared to the way we scan. And what we're seeing actually is these red lines. And these red lines do not exist in the sample. So we're creating artifacts. But on top of that, in the middle here, we do not detect this at all because we don't measure it. Okay? So the red line are detected, they do not exist. But the green line are not detected and they are in a sample. So we're creating artifacts and we're missing information when we undersample on a single point confocal. So what I have said so far was sampling in XY. Now what happens in Z? When we acquire Z stack on a microscope, all the microscopes are equal. That means we always acquire Z stack in the same way, regardless of whether we acquire Z stack on a single point confocal or a camera-based system. The way we acquire the Z stack is that we move the objective to a different plane and then we take another image. Or otherwise we move the sample to another plane and then we acquire an image. So we take an image and then we move and then we acquire an image. So it's exactly the same thing as when we acquire on a single point confocal, we basically do not measure between the planes. So when we take a Z step, this is the step between the different planes. So the distance between two images in a Z stack is the Z step. And there is a magic button, so we can use the magic button, which can be called Nyquist or recommended Z step. So it has different names depending on the company, but it's the same thing. And what it does is that it sets the Z step to the to fulfill the Nyquist sampling criterion. So what is that? It's the smallest distance resolved in Z by the objective. So the Z optical resolution must be sampled more than twice. It's exactly the same. It's the same for everything. So it's very convenient. So here is the optical resolution in Z. Equation is 2n lambda divided by ns squared. And this is the radius of the main lobe of the point spread function in the axial dimension. So in this, in Z. That is the optical resolution. And what we need to do is we need to put two Z steps in that radius, okay? So we have a 2 here. So the Z steps, we can just remove the 2 and then we know the size, the maximum size of the Z steps. So these Z steps should be that distance apart or smaller steps. We can go smaller as well, but then we oversample. So when we scroll through a Z stack that you have acquired, the significant features in the image, and that is up to you to decide what the significant features are, because it depends on your scientific question. So the significant features should be visible in more than 2 planes. So I show you what I mean. Here I have a Z stack, which is very highly sampled, so I have small distances between the Z planes. If you focus on one of these dots, you will see that each dot appears in several Z planes. So you can see the dot for quite a while, and we keep on going from one plane to another, and you will see that each dot is represented in more than 2 images in this Z stack. And that means that I am actually I am sampling these dots in the correct way. They must be represented more than 2 images. Now, if I instead use very big steps, I do not measure between the Z steps, right? And you check one spot, each of them is only represented in 1 image in the Z stack. That is undersampling these spots. Not only you will create artifacts, but also you may miss some spots, okay? So this is the way you check with your scientific question if you're sampling enough or not. So the signal between the Z planes is not recorded. And when you undersample in Z, you also create artifacts in the reconstructed volume, and you're missing information because the signal is not recorded in between. So this was in Z. Now, you guys take your calculators and we're going to do a little exercise. So what are the image pixel size in XY and the Z step that fulfill the Nyquist sampling? We have these two, so we have 2 objectives, 60x NA 1.49, and we have 40x 0.95. This is an air objective, so the refractive index of the immersion medium is air, so it's 1. And this one is an oil objective, so the refractive index of the immersion medium is 1.52. And we're imaging EGFP, so the emission wavelength is this. What is the optical resolution for these 2 objectives in XY? So it's 0.61 lambda divided by NA. Anyone can tell me? So what is the optical resolution of the 60x objective?
208.
208. Everybody agrees? What about the 40x? Again, 0.61 lambda divided by NA. This is lambda. This is NA.
333.
Okay, so 200, you said 208. Okay, that was a bit funny. And 334. It depends how we round up. So this is what I got. Maybe I'm not sure. I may have made a mistake as well. Which image pixel size will fulfill the Nyquist sampling criterion? How do we figure out that? So the size of the image pixels, or Z step, should be half of the optical resolution or smaller.
So we can say 106. Like it should be smaller than 106.5. So
yes, 106. Okay. All we do is we take this guy, we divide it by 2, and we say this is the maximum size. So it should be smaller than that. Okay. What about the other one?
167.
167. Good. So we just take this one, the optical resolution divided by 2, and this is the maximum size. They can be smaller, but they cannot be bigger. Otherwise, they do not fulfill the Nyquist sampling criterion. How about Z? So the Z resolution is 2n lambda divided by n square. We have n here, we have a lambda in here, and we have na here.
712.
712. Good.
Oh,
And the other one? Anyone? Did you find Chen?
It's 1152.
Bingo. How about that? What is the Nyquist Z step? The maximum Nyquist Z step. So the maximum distance we can have between two Z planes in our Z stack.
356.
Yes. Good. Easy. We just divide by 2. Okay? And this is the maximum. So we can have smaller, but not bigger than that. Well done. So you see, it's very simple, really. It's a pretty simple, but it's very important to understand. So I'm going slowly to explain. On the single point confocal, how can we set the pixel size to fulfill the Nyquist sampling criterion? You guys tell me. What can we do on a single point confocal if we want to set the pixel size, the image pixel size to Nyquist?
For a size system, we put the optimal button.
Click on the magic button. Very simple, okay? All right, and then on a camera-based system, how can we check if the image pixel size fulfills the Nyquist sampling criterion? How do we know? So we know that this is the pixel size we should have in the image, but how do we know what the image pixel size is?
We divide the camera pixel with the magnification.
Yes, we can do that. And we can also check in the software. So the image pixel size is going to be the camera pixel size divided by the magnification. And that number that we get should be smaller than this. They should be smaller than half of the optical resolution. Okay, so it's quite simple. But in this case, you need to know the camera pixel size, which you will know very soon because you will have to check. And then we can also look in the software because the software will tell us the size of the image pixels. And that's also quite simple. So now, how can we set the Z step so that the Z stack fulfills the Nyquist theorem, regardless of the system? On any system, how can we get the Nyquist Z step?
We have a button too.
Push the button. I like that. Magic button. It works for all types of microscopes. Well done. Job. So I think you got the idea. Now, there's one more little layer of complication, let's say, is what do we actually need? Because now we see that the objective is resolving something, and then we are now digitally resolving it if we are Nyquist. But does that correspond to what we really need for our scientific question? And that's also very important. That's actually the key. So, oh yeah, I forgot one thing, sorry. Before I go to that, we're going to talk about time lapses. When we image a time lapse, how often do we sample? How often do we measure and do we acquire an image? This is a very important question. And clearly, when you image a time lapse, it's likely that you have a live sample. And therefore, you want to be very restrictive with how often you image. So how do we do this for time lapse? Well, the Nyquist theorem works there as well. So it's brilliant. And basically, you have to decide, based on your scientific question, you have to decide what is a significant change for your scientific question. When you determine what is a significant change, then you divide this by 2, and then you have the time interval. So here is an example. We have beating cardiomyocytes, and this is what they look like if my movie starts. Yes, so this is what they look like, and they are pulsing, and we have a calcium sensor in there, so we can see the calcium pulses. So in here, I have a 20 millisecond interval, so I am imaging very, very often. I'm imaging 50 times per second, so 20 milliseconds in between the images. And let's say I have 2 scientific questions. One of them is there a pulse within this time frame, within the time I'm recording the whole movie? Is there a pulse of calcium or not? And the second scientific question is, where is the peak of the pulse, right? Because it goes up slowly, and we can say here maybe is the peak, and then it goes down, right? So I have these 2 scientific questions. Now, if I instead record every 200 milliseconds, so instead of having 50 images per second, I have 5 images per second. And you can see what the images look like. I have a lot rougher readout of what's happening in my sample. One, if we know that the pulse is longer than 2 times 200 milliseconds, then this is enough, because we can see that there is a pulse, right? But this is as long as the pulse is longer than twice our interval, then I know that this is it. So you have to sort of include, like almost for everything in microscopy, you need to include your knowledge of the sample in your decisions. So we have here, if the pulse is longer than 2 times our interval, then we're fine with this. But if we want to know where the peak is within the pulse, you can see that we do not have a fine enough readout here compared to this one, where we had a very, very fine readout. We can know exactly where the pulse is, okay? So if the significant change are faster than 2 times our interval, then this is not accurate enough. So we are undersampling here to answer this question, but we're sampling enough to answer this question. So it's all related to the scientific question, and you have to figure out what is the significant change in the image. So in here, when we look at this one, we can see that from one image to the next, we have very, very small changes, but we have a very fine readout. So we can see the significant change. We divide by 2, and this is what we want. So this means that in this image, we may oversample. Maybe this is oversampled, and it leads to light toxicity for this live sample. So we also don't want that. It's very important. So in the Nyquist theorem for time, what we need to do is figure out how long it takes to have a significant change compared to our scientific question, and then you divide this by 2, and this is your time interval. Okay, you should have 2 time points in the significant change. So is undersampling ever acceptable? Is it okay to undersample ever? sometimes in our biological experiments, we're limited by some things like light toxicity for live samples, or some objective properties. For example, if we have a model, we maybe need the working distance of the objective, and maybe we'll have to pick one objective and one type of system, and that will force us to undersample. Is that okay or not? You need to realize that only the details that are undersampled create the aliasing artifacts. So the details are undersampled. It means that they are resolved by the objective, but not resolved by the digitalization. So the image details that are well sampled are corrected. So within one image, we will have some areas that do not create artifacts and some areas that will create artifacts. So you can see in this image that we have some coarse hair in here. And these are well sampled. So we do not need a fine digital resolution for these guys. The objective resolves everything in this image, not everything. It resolves this area and this area. But here we are sampling according to Nyquist in this area. However, in here, so we do not create any artifacts in here. However, in here, we can see the moiré artifacts, and that is because in this area, the objective resolves these details, but not the digitalization, so not the camera. So only the areas in the sample that are resolved by the objective, but not by the digitalization, will create artifacts. As we get closer and closer to Nyquist, so if we get smaller and smaller pixels, we are creating less and less artifacts in the image. So we have less and less aliasing artifacts. We have more accurate information in the image, and we miss less information when we record on single point confocals at stack or time. And so you can see that there is a gradient of. It's not like I'm undersampled and I'm oversampled. This is like a gradient. So what is acceptable or not acceptable? How good is good enough? And the choice of objective and sampling must be driven. So both the objective and the sampling must be driven by a well-formulated scientific question. So everything comes down to what is your scientific question? What do you need to be accurate in the image? And what is okay? If you want to count in this image, if your goal is to count the number of hats, no problem. If your goal is to measure the distance between here and here, no problem. Okay, but if your goal is to try to find some fine information in this structure here, for example, all these folding here, then you're going to be fooled by all these extra things that come onto the image. Okay, you may think that this is folding as well. So it all depends on what you want to measure, and we'll all come down to well scientific question. You must define the quantifiable metrics, so distance, volume, time, intensity, that need to be resolved. So they must be resolved, sampled correctly. And the things that you don't care about, then it's okay to undersample. Okay, so you have to consider your knowledge of the biology of the sample and how large the expectations are. The variations are between the samples. So if you want to measure very fine changes between your sample and the treated and the control, for example, if you want to measure very fine changes, you must be extra super careful with the digitalization and everything else. If you want to measure big differences, no problem. Again, you get away with murder. Okay, so the smaller the differences, the more accurate the analysis must be. And therefore, you need to have an excellent sample and excellent imaging, including the sampling. So if you take an overview showing large image areas of interest, it's totally okay to undersample like heavily. If you image sparse nuclei that need to be counted, you can undersample. It's not a problem. But if you are looking at small crowded objects, if you undersample, then you will create objects and you will create artifacts in the image that will influence your results. If you're not sure, what do? You take a little time lapse of 5 undersampled images, like the way you would like to sample them, for example. You take 5 images, and then you take 5 images of the same image, but acquired with Nyquist sampling, and then you run through your analysis, and you see what the differences are in the result. Okay. So this way you can see if the undersampling and the artifacts that you're creating in your image influences the results that you have. I look at what my time thing. I think I'm good. Okay, so here are the conclusions for this lecture, where we spoke about contrast and sampling. If you assume a perfect sample, and you know how easy it is to be far away from a perfect sample, so if we assume a perfect sample, then the resolution of the optical image, so the resolution of the image that the objective creates, is limited by the size of the point spread function. Okay, so the diameter of the main lobe, the radius of the main lobe. It is a perfect sample, and we sample correctly. The way we record we digitalize this optical image can sometimes limit the final resolution in the image, and in that case, we are undersampling. On all systems, the undersampled details in the image are incorrect, and we're creating artifacts. This incorrect information in the intensity and in the shape and in the number if they fuse. So we create aliasing artifacts, lines that do not exist, incorrect intensities, round objects that become elongated, etc. On single point confocals, in Z stacks on all systems and in time lapses on all systems, on top of creating aliasing artifacts, we also stop recording some information. So the signal in between the sampling points is not recording at all, and therefore we have also missing information, not only artifacts, but also missing information. So how do we avoid undersampling? The distance between the XY optical resolution must be sampled, or the Z resolution must be sampled by more than 2 image pixels or 2 Z planes in Z. Okay, this is what the Nyquist sampling theorem is. So we must cram 2 pixels in this distance of the optical resolution, the radius of the PSF. So in XY, the image pixel must be smaller than half of the XY optical resolution, and in Z, the Z step must be smaller than half of the Z optical resolution. On single point confocal, there is a magic button that will set everything to the correct sampling. And on camera-based system, we must check that the pixel size is half the XY optical resolution or smaller, and we do that by calculating the camera pixel size. We divide by the magnification, or we look in the software what the image pixel is. On all systems, there is a magic button to set Z. Okay. And the Nyquist sampling also works with time. So the time interval in a time lapse should be half of the time needed for a significant change to occur, or it should be shorter. And what the significant change to occur is for you to decide when it's going to be preventing you from answering your scientific questions or not. That's what it means, right? Whatever you need to measure to make sure you answer the scientific question, this is the time for a significant change. You divide that by 2, and this is your time interval. If we undersample, anyway, for whatever reason we undersample, then the image resolution is the size of 2 image pixels, and in Z is the size of 2 Z planes. Okay, so, sorry, I didn't write it properly. I should not have written like this because it looks like it's divided. That's not the case. It's in XY, it's the size of 2 image pixels, and in Z is the size of 2 Z planes. And that is true for all systems. So we need to reverse design our experiment. First, we define our scientific questions in terms of which quantifiable metrics we need to resolve. And then we decide how much resolution is needed and which differences we need to be able to reliably interpret. And this deducts, and from there we figure out which microscope we need to use, which objective, and which sampling Okay, so we start from the scientific question and then we decide how we design everything. And on top of that, we try to make our sample perfect. So that is it for this lecture. And I would like to hear what you think. Did that come as a surprise?